Every single living thing, whether it's a tiny bacterium or a massive blue whale, starts its life as just one cell. So how in the world does that one cell turn into trillions? Well, the answer is a process so fundamental it's literally the engine of life itself, cell division. So today, we're going to dive into how this incredible process allows us to grow, to heal, and even to create the next generation.
So think about it. Life has two massive challenges to solve. First, how do you build and repair a single complex organism that has trillions of cells that all need to be identical? And second, how do you create enough genetic variety to make sure a species can adapt and survive over time? Turns out, it's all done through two different, absolutely brilliant strategies for cell division.
All right, here's the game plan for today. We're going to start with the blueprint of life itself, the chromosome. Then, we'll get into the body's biological copy machine, a process called mitosis. After that, we'll look at how life shuffles the genetic deck with meiosis. And finally, we'll put them head to head and see why life absolutely needs both.
Okay, so before a cell can even think about dividing, it has to do something incredibly important. It has to make a perfect copy of its entire instruction manual. And all of those instructions are neatly packed into a structure called the chromosome. The best way to think of a chromosome is like a super tightly wound up book in a massive library. Each one of these books contains a volume of the genetic instruction manual for building and running an entire organism, and all of this precious information is packed safely inside the cell's command center, the nucleus.
Now, when you picture a chromosome, you probably think of that classic X shape, right? But it's not always that simple. Their shape actually varies, and scientists classify them based on the location of this little pinch point called the centromere. When it's smack dab in the middle, it's called metacentric. But if that centromere is a little bit off-center, creating one short arm and one long arm, well, then we call it submetacentric. When it gets pushed way up near the end, creating a really short arm and a very long one, that's called acrocentric. And if it's right at the very tip, it's telecentric. So why do these shapes even matter? Well, they're like genetic fingerprints, helping scientists identify and sort our chromosomes during analysis.
So, with our blueprint understood, let's look at strategy number 1, mitosis. And honestly, the best way to think about this is as the body's biological copy machine. Its one and only goal is to make perfect, identical clones of cells. It's the reason a little cut on your finger heals, and it's how you grew from one single cell into the person you are right now. So if there's one thing to remember about mitosis, it's this. One cell divides just once to create two identical daughter cells. These new cells are perfect genetic copies of the original, containing the full set of chromosomes. In humans, that's 46. And the scientific term for having a full set is diploid. But this whole thing isn't just a chaotic split. Oh no! It's this beautiful, carefully choreographed dance that happens in 4 main acts. So let's walk through them.
The process kicks off with prophase. This is where the cell's DNA, which is usually like a messy bowl of spaghetti, starts to coil up super tight. It becomes those condensed, visible chromosomes we can actually see under a microscope. At the same time, the wall around the nucleus dissolves, basically clearing the stage for the main event.
Next is metaphase. A super easy way to remember this one is M for middle. That's because the chromosomes, which have already copied themselves, line up in a perfect single-file line right down the equator of the cell. It's all very neat and tidy. And then, bam, anaphase. Think A for away. The cellular machinery grabs each copy of the chromosome and yanks them apart, pulling them to opposite sides of the cell. In that instant, each side of the cell now has a complete and identical set of genetic instructions.
And finally, we get to telophase. This is the grand finale. 2 brand new nuclei form, one around each set of chromosomes. The chromosomes start to uncoil back into their stringy form, and the cell membrane pinches in the middle until, poof! Where there was one cell, there are now 2 perfect clones.
So that's our copy machine. But life isn't just about making perfect copies, right? It also needs a way to mix things up, to create variety. And that's where our second strategy, meiosis, comes into play. This isn't about cloning at all. It's about shuffling the genetic deck to create totally unique reproductive cells.
Now, meiosis is a whole different ballgame. It involves 2 rounds of division, not 1. And the result? 4 daughter cells, not 2. But here's the real kicker. These cells are all genetically unique, and they only contain half the number of chromosomes from the parent cell. We call this haploid. And these special cells are, of course, the gametes, sperm and egg. So where does all this amazing variety come from? Well, the real magic of meiosis happens right at the beginning, in a super complex stage called prophase 1. This is where the chromosomes you got from your mother and the ones you got from your father actually pair up and physically swap pieces of their DNA. It's an incredible process called crossing over, and it happens in this carefully orchestrated 5-step dance.
Okay, here's how it works. Your homologous chromosomes, like, say, the chromosome 9 you got from your mom and the chromosome 9 you got from your dad, they find each other inside the cell. Then they literally trade genetic information, swapping little segments of DNA. This is crossing over. It's like taking 2 different decks of cards, cutting them both in half, and swapping the bottom halves. You end up with 2 completely new, totally shuffled decks.
So the entire goal of that first big meiotic division is just to separate these homologous pairs. Unlike in mitosis, the copied chromosomes, the sisters, actually stay together. The cell just pulls the entire maternal chromosome to one side and the paternal one to the other. This is the key moment where the cell goes from being diploid with 46 chromosomes to haploid with just 23.
After that, meiosis 2 begins. And you know what? This second division is basically just a round of mitosis. In each of the 2 new cells, the chromosomes line up in the middle, and this time, the sister chromatids get pulled apart. The final result? 4 unique haploid cells, each one ready for reproduction.
All right, we've walked through both of these amazing processes, so now let's put them head to head to really make the differences crystal clear.
This table really just nails it, doesn't it? Mitosis is for growth and repair. It's one division that creates 2 identical diploid cells. Meiosis, on the other hand, is for sexual reproduction. It takes 2 divisions to create 4 genetically unique haploid cells, all thanks to that amazing crossing over. 2 completely different outcomes for 2 very different biological needs.
So we have these 2 strategies, mitosis for uniformity and meiosis for variety. You can think of it like this. One is working for the individual organism, and the other is working for the survival of the entire species. Let's break that down.
Mitosis ensures the stability of you, the individual. You absolutely need every cell in your body to have the same reliable blueprint to function properly. But meiosis ensures the survival of the species. By creating all this genetic variety, it gives a population a bunch of different tools to survive disease, climate change, or any new challenge the world throws at it. It's the ultimate evolutionary insurance policy.
Which brings us to a final, really fascinating question. Making simple copies is easy and it's efficient. Shuffling genes is complex and it's costly. So why has nearly all complex life on Earth adopted this more difficult strategy? The answer speaks to the profound evolutionary power of variety and to the endless, beautiful dance between stability and change.